Assalamu alaikum everyone. I hope you're all having a great day. My name is Mohamad Behairy. I am a third year medical student. Inshallah, today we will be explaining and going through two of your MOL lectures that are required for the midterm. The first one is nucleic acids and their structure, or the second one is DNA replication. بإذن الله, they are very easy lectures. This PAL session will include literally everything you need to know. All of the practice questions in this PAL session will be the same questions, inshallah, that you will see in your exam, both the midterm and the final. But let's not waste more time. Let's begin. طيب, we will start with nucleic acids, the first portion of this lecture. We have to establish a very core and important concept. تمام? In our body, whenever we want to synthesize or create any protein, there has to be a list of steps that we have to go through and do before we can get to this final result of a protein. And this whole step and process is referred to as the central dogma of biochemistry. It explains the steps taken to achieve this final goal of having a new created protein. It starts all the way from the DNA level. Allah yahayik. It starts all the way from the DNA level. Well, this DNA will replicate itself. We want to have more and more DNA so we can have more and more cells. Well, this DNA will become RNA through a process called transcription. Well, I'm sure you guys have lectures on the process of transcription. Well, after that, this RNA will then be translated into what we call proteins. فإحنا today in this lecture, we will cover everything related to DNA or how this DNA replicates. I just want you to know that this whole process is called the central dogma. طيب, now let's talk about this DNA and RNA. Let's talk about them in detail. What do we refer to when it comes to DNA, RNA? We call them nucleotides. A DNA is a nucleotide, an RNA is a nucleotide. تمام? A nucleotide is a structure that has a specific composition of substances. Yani in every nucleotide, you will find the same structures making this nucleotide up. Let's break this down into detail. تمام? Every nucleotide will start by having a phosphate group, a simple phosphate, a P. Other than the phosphate group, it will also have a nitrogenous base. Now, what is a nitrogenous base? We will talk about them in detail, but I'm sure you guys recognize them. The adenine, thymine, guanine, cytosine, ATGC. These are called nitrogenous bases. They are simple bases, structures, that are made up of nitrogen. تمام? For now, so far, we have a phosphate group and we have a nitrogenous base. Last but not least, because it's the most important one, the sugar. Well, the reason why the sugar is the most important structure in a nucleotide is because the sugar is what determines if we're going to have a DNA or an RNA. For the phosphate group, you're going to find it in all of the nucleotides, RNA or DNA. Same thing with the nitrogenous bases. You're going to find them in DNA and RNA. But the sugar, some certain kinds of sugar will give us DNA, while others will give us RNA. Well, now we're going to talk about the different types in a bit. But from this slide, I want you to remember, this is very important, guys. You will be asked about this. Any nucleotide is composed of three important things: a phosphate group, a nitrogenous base, and a sugar. So for those of you who prefer text, the central dogma, it describes the process of DNA replication into RNA transcription, وبعدين we translate this RNA into proteins. Well, a nucleotide, as we said, it consists of three things. One, a phosphate group, two, a nitrogenous base, and three, a sugar. Well, as we said, this nucleotide can either be a DNA, a deoxyribonucleic acid, or a ribonucleic acid without that deoxy, which is going to be RNA. Now let's take a look at this sugar. مو احنا قلنا that this sugar is what's going to tell us if this is a DNA or an RNA. But let's take a look at the sugars. Well, let's try to differentiate. طبعا from the word ribose, you know that this is RNA لأنه ما في deoxy. Well, here it's written deoxyribose, but this is DNA. But let's take a look at the sugar itself. I want you guys to do and follow these two steps whenever you want to identify if it's a DNA or the RNA. Step number one, go to the second carbon. This one, carbon number 2. The second step, look at what's attached to this carbon. If it's an OH, a hydroxyl group attached to the second carbon, then we have an RNA. If the second carbon has an hydrogen ion, an H attached to it, then this is going to be a DNA. This is exactly why I said that the sugar is very important. just as I said a few slides ago. And depending on what this sugar is made of, we're going to have either the DNA or the RNA. So this is a short table that basically tells you all the differences you need to know. RNA has an OH at the second carbon, which is here, as you guys can see. This is carbon number 2, this is the OH. While a DNA would have a hydrogen as the second carbon, or attached to the second carbon. An RNA, you guys know when a DNA are double-stranded, they are a double helix shape. لا, بس ال RNA, it's a single strand. RNA is one strand, DNA made up of two strands. تمام? Well, do you guys remember when we talked about the nitrogenous bases here early on? We said in any nucleotide has a phosphate group, sugar, and a nitrogenous base. There are certain خلينا نقول types of nitrogenous bases. I talked about them قبل شوي adenine, guanine, cytosine, thymine, تمام? All of these, they're important. They make up the DNA or the RNA. There is just one difference, تمام? In DNA, we have thymine. But in RNA, we don't have this thymine, which will be replaced by what we call as uracil. We're going to talk about these nitrogen bases in detail in the slides. طيب, أول شيء, let's start by agreeing إنه a nucleotide is made up of something called a nitrogenous base. You need to know that there are two types of nitrogenous bases, purines and pyrimidines. تمام؟ يعني any certain nitrogenous base, this base in itself, it can either be a purine or a pyrimidine. But what's the difference between a purine and a pyrimidine? What makes them different? The purine will have multiple rings, two rings. You guys can see here, this is one ring, one molecule. Or attached to it is a second ring. If you guys see something that looks like this in the exam, where we have two different rings, then this is immediately a purine. طيب, what if we only have one ring, زي كده, only one thing, nothing attached to it, then this is what we call a pyrimidine. تمام? Or remember how we talked about adenine, guanine, cytosine, thymine, uracil? Those five, they classify into either purines or pyrimidines. Sorry, what is the difference between those two? Okay, I'll repeat. Look, focus with me. In a purine, look at the structure. We have one pentagon and next to it another pentagon. But in biochemistry, we don't say it's a pentagon. We call it a ring. So in a purine, we have two rings, one ring over here, and attached to it is another ring. But in pyrimidines, you're only going to see one ring, one pentagon, nothing attached to it. Or, yeah, it's a pentagon, nothing attached to it. When the exam, they could easily get you this picture on its own, and they would ask you, is this a purine or a pyrimidine? So you would see, no, there are two rings. Because there are two rings, we would call this a purine. If it was one ring, it would be called a pyrimidine. Is that clear? And about the subtypes we talked about, adenine, guanine, cytosine, all of those, we classify them into either purine or pyrimidine. So an easy way to remember this is purines. Purines are pure. What is also pure? Gold. So purines are pure as gold. P for purine, A for adenine, as G for guanine, gold. But this is an easy way to remember which nitrogenous bases are purines. When will this get uploaded on YouTube? Probably after this file session and ends by like 2 hours, inshallah. But let me repeat, an easy way to remember what nitrogenous bases are purines is by remembering this mnemonics. Purines are pure as gold. P for purines, A for adenine, as G for guanine, gold. تمام? That's how you can know adenine and guanine are purines. Well, this means that anything else, which is cytosine, uracil, and thymine, they're going to be pyrimidines. Okay? Well, it's very important to know which ones are purines and which ones are pyrimidine. So remembering the pure as gold mnemonic makes things much easier for you to remember. Well, as we said in the previous slide, uracil is only found in RNA, while thymine is only found in DNA. This is very important to remember. All of the rest can be found in both DNA and RNA. تمام? Now, let's move on to a very important concept. Do you guys see these nitrogenous bases? They actually bind to each other, but not all of them like each other. So adenine could bind to one of them, but not the other. Depends on who they like, basically. And you guys have to memorize what each one of them likes. But fortunately, it's very simple. طيب, before we get into this, adenine, guanine, cytosine, and thymine. Which two of them start with a similar letter? It's guanine and cytosine. The letter G is very similar to the letter C. It's only this dash at the end that's the difference. So that is why, because they're very similar, guanine and cytosine will be the ones to bind to each other. Whenever we have a cytosine molecule, guanine will bind to it. Whenever we have a guanine nitrogenous base, cytosine will bind to it. Nothing else can bind to it. Those two only bind to each other. تمام? وبعدين we have the rest, which are adenine, uracil, and thymine. Adenine will bind to thymine in DNA or to uracil and RNA. تمام? Oh, you also remember it like that? That's amazing. I think this is the easiest way to remember it because G and C look very similar. They're very similar letters, letter G and letter C. So using that link, you can remember in guanine and cytosine will be the ones to bind to each other, while adenine, uracil, and thymine, the other, which are the rest, will also bind to each other. تمام? Oh, guys, please remember, pure as gold, purine, adenine for A, and guanine for gold, which is G. طيب, at AU as well as basically as in Al Faisal University. Oh yeah, I never thought about that, but yeah, what she's saying is adenine, uracil, Al Faisal University. That's an easy way to remember it too. Yeah, صراحة, it's very easy to make mnemonics for these. But if you know guanine or cytosine, خلاص, everything else will be together. Adenine will bind to uracil and RNA, and adenine will bind to thymine in DNA. Now, guys, the last concept from this whole slide, something very, very important. You guys have to remember it very well. احنا didn't we say إنه guanine binds to cytosine? طيب, there is going to be a number of bonds linking this guanine and cytosine together. You have to memorize what the number of this bond, or how many bonds do we have. Between guanine and cytosine, we're going to have 3 hydrogen bonds. You have to memorize this. If you have G and C linked together, this means that they have 3 hydrogen bonds. You have to remember this. While adenine, uracil, or thymine, when they bind to each other, they only have 2 hydrogen bonds. Well, a better way to demonstrate this is here. You guys can see this is adenine. Well, obviously, adenine, pure as gold. What is adenine? A purine. That's why it has two rings. You guys see that? While thymine, it's not part of the pure as gold. That's why it's a pyrimidine which has one ring. Just wanted to revise this concept. طيب, فبetween adenine or thymine, how many bonds did we say exist? 2 bonds. This one, the first one that's here with my cursor, with the second one that's here. While between guanine or cytosine, again, guanine, pure as gold, G for guanine, it's a purine, so we have 2 rings. And cytosine, not pure as gold, so it has one ring, it's a pyrimidine. Between the G and C, we're going to have 3 hydrogen bonds. I want you to remember this. So for those of you who prefer text, just to summarize, nitrogenous bases can either be purines, which have 2 rings, or pyrimidines, one ring. Purines, as we said, pure as gold, adenine and guanine, while pyrimidines are everything else, cytosine, thymine in DNA, and uracil in RNA. So the base pairings, which nitrogenous bases come together, as we said, A and T, or U in RNA, and this consists of 2 hydrogen bonds, as you guys see here, and C going to G. Look, guys, how similar the letter G is to the letter C. But no wonder they would bind together. And between those two, we would have 3 hydrogen bonds. Tamam? Now, last but not least, I want you guys to remember this. It's a very simple concept, very easy, but I just put it for the sake of completion. Obviously, we have adenosine triphosphate, adenosine diphosphate, and adenosine monophosphate. Which one of them has the highest amount of energy? Obviously, it's the adenosine triphosphate because there are 3 phosphates, which is going to have more energy than the one with 2 phosphates, which would have more energy than the one with 1 phosphate. This is a very random concept. I just want you to remember that it's very logical. It can easily be deduced by common sense, but I just put it for the sake of completion. Now, let's take a look at the structure, tamam? There's something weird about the structure. At first glance, you might say, oh, this is just a random nucleotide. We have the sugar, we have the pyrimidine in this case, or here, مثلا, it's not a pyrimidine. We have two rings, so it's a purine. But don't you guys think there is something missing in this nucleotide? I want someone to tell me in the chat right now Out of the three components of a nucleotide, what is missing here? Exactly, the phosphate group. يعطيكم العافية. So this is a nucleotide at first glance. You can see the sugar. Here in the number two, carbon number two, you can see OH. Okay, yalla, another question. If we have OH at the second carbon, what is it going to be? RNA or DNA? RNA, ya salam alaikum. Absolutely. We can also see this ring. But is this a purine or a pyrimidine ring, a nitrogenous base? Is it purine or pyrimidine? Pyrimidine, absolutely. طيب, as we said, what's missing is the phosphate group. We actually have a name for this structure where we have the sugar, we have the nitrogen base, but we're missing the phosphate. We call this a nucleoside, not a nucleotide with a T, a nucleoside with an S. It's basically a sugar and a nitrogen base without the phosphate. Tamam? Just remember it. They could easily ask you a question. They could give you, for example, this one, the one I'm pointing at with my cursor. They could ask you, is this a purine nucleoside or a pyrimidine nucleoside? You would obviously be like, it's a purine nucleoside, not tide. Why? It's a nucleoside because it doesn't have a phosphate, and it's purine because the nitrogen base has not one, but two rings. Is it abnormal? No, no, it's not abnormal. It's simply a kind of structure that is found in our body. Nothing abnormal about it. This nucleoside could one day have a phosphate attached to it, and it could become a nucleotide. Nothing abnormal about this. Right. But as we said, nucleoside means it has everything except a phosphate. Is inosine a purine nucleoside too? This one? Yes, it is. Inosine, you can see the nitrogen base has two rings, so it's a purine, and we don't see a phosphate, so it's a nucleoside. Absolutely. Like, of course. Moving on to Chargaff's rule. This is one of the most important, but honestly stupidest and easiest rules in all of medicine. It basically says that the bases that bind together, we said A binds to T and C binds to G. Any bases that bind together and like each other have to be in the equal concentrations or equal amounts. Yani if we have 10 adenine, we're going to have 10 thymine because they bind to each other. If we have 30 million guanine, we're going to have 30 million cytosine. You guys can see in this table, it's basically an experiment, which shows you the amounts of adenine are always pretty much the exact same as the amounts of thymine. Why? Because they bind to each other. Well, I want someone to remind me, how many hydrogen bonds do we have between adenine and thymine? Okay, no, not three, two. Adenine and thymine, two, yes. But what about guanine and cytosine? Exactly, three. يعطيكم العافية. And obviously, this rule also applies to guanine and cytosine, where basically we have similar amounts of guanine to the amounts of cytosine. They're all equal, tamam? But for those of you who like text, Chargaff's rule simply states that there are equal concentrations of bases that pair with each other. But the percentage of adenine always equals the percentage of thymine. The percentage of cytosine always equals the percentage of guanine. But the way they're going to ask you this in the exam will be a two-step process. Let's take this question as an example. It's telling you that a sample of DNA has 10% cytosine. What is the percentage of thymine? It's not asking you for its opposite, which is guanine, or who it pairs with. It's asking you for something completely different. So it's going to have to require some math. Absolutely, 40% is the correct answer. Why? I'll explain why. We have 10% of cytosine. What does this mean? We're going to have 10% of guanine, a 10% of cytosine, and 10% of guanine. That's 20% gone. How much do we have remaining? 80%. This 80% will be shared between adenine and thymine. صح? طيب, we said in this rule states that adenine and thymine, they have to be in the exact equal, let's say, concentrations. So if we have 80% left, we're going to have to divide it by 2. 40% for adenine and 40% for thymine. That's why the answer to this will be 40%. Is that clear, guys? Then you will definitely get a question about this, يعني, inshallah. It's very easy, an easy grade, basically. طيب, one last thing I want you to remember, this rule does not apply to viruses. So if you get a question in the exam, it gives you very random percentages of adenine or thymine or guanine, very random percentages. This means this is a virus. If it's a living thing, a bacteria, a human, or whatever, they have to be the same. Adenine, thymine, guanine, cytosine. If they're different, it's a non-living thing, most likely a virus. طيب, moving on to the first checkpoint. Does anyone have any questions so far? Do we just assume it's a virus? Yes. If the rule doesn't apply if it's single-stranded, right? Yeah, obviously, because this has to be double-stranded, where the A from one strand applies to the, or binds to the T from another strand. وأصلا if it's one strand, it's no longer technically DNA, it becomes RNA. This is a rule for DNAs. تمام? That's number one. Number two, I just want you to remember, and when it comes to viruses, you can see 20 adenine but 60 thymine. You might be like, wait, aren't they supposed to be equal? لا. Can you repeat how you get the thymine? Absolutely. طيب, let me use a pen. طيب, we said here in we have 10% cytosine, صح? طيب, I want you to tell me, Lamar, if we have 10% cytosine, how much guanine are we gonna have? Because we said some certain bases should have equal concentrations. 10, absolutely, 10. So 10% cytosine means that we will also have 10% of guanine. طيب, so that's 20% خلاص, gone. We know what it is for. 10% of it cytosine or 10% guanine. طيب, now we have 80% remaining. صح? 80 out of the 100%. This 80 will be shared between adenine and thymine. طيب, how much do you think each one of them should have in terms of percentage? If we said in a Chargaff's rule says in adenine and thymine equal each other, with cytosine and guanine equal each other. Exactly. So we will have 40 of this and 40 of that. خلاص, that's pretty much it. Of course. What about uracil in Chargaff's rule? Is it also equal to adenine? Yes. 100%. 100%. بس احنا we're talking about DNA. Guys, this concept in the exam, it will be only tested with regards to DNA. This is a lecture about DNA and DNA replication. With RNA, you have to worry about the transcription, translation, these kinds of stuff. But yes, uracil is basically thymine, guys. It's just it's an RNA. طيب, طيب. So let's move on to the first question I have for you guys. Let me know what you guys think the answer is in the chat. The percentages are a bit confusing, but A is 20%, T is 25%, C is 30%, G is 20%. But basically, this question is saying if A has the complement of T, how much T are we going to have? If T has a complement of A, وهكذا. Okay, we have three answers so far. Yalla, a few more answers, guys. It's a very easy question. I just want you to tell me what binds to T, for example, and obviously it's going to have the same percentage when it binds to it. Exactly. The answer is A. The answer here is A, not B. Look, let me tell you why. We said, we have 20% A, صح? Well, here they were asking about the complementary strand. The complementary strand is basically the strand that has bases that can bind to the original strand. But in the original strand, we have A. But in the complementary strand, we would have T. Then T is what binds to A. Well, because A was 20% originally, this T will also be 20%. In the original strand, we have T, which is, T, 25%. What is complementary to T? A. What is A going to be in this case? وهكذا. تمام? And sorry, here A is 20, T is 25, C is 30, and G is 20. But what you guys said is correct. A is the answer. Is this concept clear, guys? Any misconceptions? Okay. You thought that if the values are random, it would be a virus. But look, there is an important distinction to be made. This is a DNA molecule, تمام? We have this strand and we have this strand. يلا guys, I'll explain the concept again من الأول. Exactly, simply switch the numbers. شوفوا. If we have 20% here, A, we're gonna have 20% here, T. If we have 40% here, G, we're gonna have 40% here, C, and so on. تمام؟ فإحنا when we're guys about the virus question, just wait until the next question. The next practice question, you'll know exactly what I mean. But leave the virus aside. I promise you'll get it. فهنا we're not comparing the values in this one strand. لا. يعني here we can have 30 thymine. It's okay. They shouldn't be equal in the same strand. They are equal in the cross strand, in the complementary strand. يعني هنا we have A20 فهنا we have T20. هنا we have T30 فهنا we will have A30. Is that clear? In what case would D be correct? صراحة almost never. Almost never would D be correct. They will always give you the data of the strand, or they would ask you for the complementary strand. You can always have a complementary strand. Right, now let's go to the next question, which all of you are asking about, the virus question. But here they're not asking you for the complementary strand. They're not asking you for the concentrations. They're just telling you, no, we have 10% A, 30% T, 20% G, or 20% C, sorry, 40% G. فهنا it would be viral genome. تمام? This is all you need to know. طبعا obviously there are details to this, why viruses have the compositions differently. But الصراحة I didn't want to go into this much detail because you genuinely don't have to know all of that. Just know in when you get this question where you have these random concentrations, it will be viral genome as the answer. Bacterial plasmid, bacteria is a living thing. We just saw in a few slides ago how they always have similar concentrations. Mitochondrial DNA, this is found in humans أصلا. Nuclear chromosome, يعني all of these things are the same أصلا. But the odd one out is viral genome. طيب, let's move on to the last question. Well, I talked about this a lot. So please, يا رب, you guys got the concept and can answer this correctly. What is the structure indicated by the image? Oh, mashallah. Everyone got it correct, mashallah. Yep, it's a C. It's a purine nucleoside. Why? Because we have a sugar. This is a sugar. We have a nitrogen base. It's purine. ليش it's purine? لأن it has two rings. صح? It's missing a phosphate. It's not a nucleotide as here. It's a nucleoside. طيب, I want someone to tell me, is this sugar a DNA or an RNA? Exactly, it's an RNA because we have an OH at the second carbon, which is here.
This is not buffers.
ممتاز. ممتاز. يعطيكم العافية guys.
I'm not watching this.
طيب, last question. Oh, this is a very good question. Well, I emphasized on this, the hydrogen bonds between A and T and C and G. So here, what you need to do, I'm just going to give you a small hint. You have to find the complementaries base of each one and add the number of hydrogen bonds between them. Yalla, let's wait for your answers.
No message me.
Let's have more answers, guys. What do you think? A is going to have a complement of T, TA, CG, GC, TA, and AT. How many bonds are between these, or between these, or between these, or between these? Just add them all up. Exactly, mashallah. Everyone answered this correctly. It's 14. Oh, by the way, guys, you don't even have to know what's complementary. A2, T2, that's 4. C3, G3. وصلنا 10. T2, A2, 2 and 2, 14. يعني you can do it easily, you know? You don't have to find the complementary base, but both ways work, obviously. طيب, now let's talk about the DNA structure. إحنا صح, we said in there is like thymine, adenine, cytosine, guanine. But how are they linked to each other? By something we refer to as a 3 to 5 phosphodiester bond. This is very important. Just memorize it like your name. How are the nucleotides of a DNA linked? 3 to 5 phosphodiester bonds. طيب. You don't have to know anything about them, by the way. You just have to know when it's 3 to 5 phosphodiester bonds. But I wanted to explain it to you a bit just so I can help it stick in your brains. But basically, any DNA strand has a 5' end and a 3' end. It's basically, what do they mean exactly? It means this is a start and this is the end of a certain strand. 5' or 3', تمام? Like a skeletal part of the DNA? Yes, exactly. But 3 to 5 phosphodiester bonds, it means the three strand, the 3' end of a single strand will make a phosphodiester bond with the 5 end of the strand of the nucleotide اللي بعده. But this cytosine, the 3 end of it, will have a phosphodiester bond with the 5' end of what's after it, which is guanine, and so on. This is all I wanted to know. After this is too much. Just wanted to remember it was 3 to 5. Oh, this is for the sake of completion. They never ask about it, but it's good to know that there are 3 forms of DNA: B form, A form, and Z form. If you had to remember something of all of this, it's that the bDNA, B, is the most common. The most common DNA is B. ZDNA is the least common. This is memorization. It's pure memorization. They're telling you that there are 3 different kinds of DNA: A, B, and Z. B is the most common, Z is the least common. That's it. Someone said they didn't understand the 3 to 5 phosphodiester bonds. True. This is a nucleotide. For example, A. This A is like this, خلينا نقول. تمام? It has 2 ends, صح? An end that is here and an end that is here. One of them, scientists decided to call it a 5' end, to the other one a 3' end. تمام? 3 to 5 phosphodiester bonds means the 3' of the first nucleotide will bind to the 5' of the nucleotide اللي بعده. That's all we're saying. تمام? That's all what we're saying. Oh, you don't really have to go into this in detail. Just remember it's 3 to 5 phosphodiester bonds. طيب, something also very important. You guys know when a DNA is like a double helix. This double helix can be, let's say, denatured, unwind, where they can separate from each other to become 2 separate strands. We call this DNA denaturation. تمام? This happens when DNA is heated. When there is a certain temperature where خلاص, one strand, one half of the DNA will dissociate from the other strand. We call this as the melting temperature. The melting temperature is basically when the temperature that's required to separate 2 DNA strands from each other. And you guys can see here a DNA with a lot of adenine and thymine at a certain temperature in degrees Celsius. خلاص وصلنا melting temperature, with the DNA strands will be split into 2. But I want someone to tell me, why is the melting temperature for cytosine or guanine, as you guys can see in the graph, much higher? Yani it takes much more temperature to melt the guanine or cytosine. Why? Exactly, because we have more bonds. We have 3 bonds as opposed to 2. خلاص يعطيكم العافية. This tells me all I need to know. You guys grasp the concept well. طبعا the 2 strands of the double helix, they separate when these hydrogen bonds are disrupted. But it's harder to separate CG bonds than the AT bonds, لأنه obviously CG are. طيب, eukaryotic versus prokaryotic DNA. There is a lot of information that you genuinely don't need about this few slides in your lecture. But I summed up the most important things about them. I want you to know, in eukaryotes, we have something called histones. In prokaryotes, we don't have histones. طبعا, what are histones? Histones are molecules that bind to the DNA. That's all I want you to know. With this complex made of DNA and histones is called a nucleosome. This is all I want you guys to know. In prokaryotes, their DNA, they're not attached to histones, ما في histones. In prokaryotes, we only have a single circular DNA molecule. It's a double-stranded circular DNA molecule. But in eukaryotes, the more complex organisms, we have the double-stranded DNAs, and we have the histones attached to them. We call this final product of histones and DNA as nucleosomes. تمام? ونيوكليوزومز are also referred to as histone octamers. ليش؟ لأنه histone octamer just refers to the amount of proteins. You don't have to go into detail. I just want you to remember what I drew. One more thing I want you to know. There is something that is associated with linking the DNA and the histones. مو إحنا قلنا إن this circular histone is going to be attached to the DNA strands. Of what links this histone to this DNA strand? It's something called an H1 protein. H1. Now this H1 is responsible for linking the DNA between the nucleosome fibers. That's all I want you to know regarding this. I'll go over it one more time to make sure I only cover what I want you to know. We have the DNA molecule in eukaryotes. In eukaryotes, it attaches to histones. How do they attach to the histones? Through the H1 proteins. All of this together is called a nucleosome. In prokaryotes, we only have the double-stranded DNA, but it looks something like this. It's a circular in prokaryotes. One more thing you need to know, in the prokaryotic DNA is very useful, لأنه we have something in them called plasmids. Plasmids help us in recombinant DNA, which basically means taking this plasmid out of the prokaryote, creating new DNA ourselves in the lab. Not a very important concept, just thought it's interesting for you to know. تمام, but these are the main differences and key things I want you to remember out of this slide. How can they ask about H1? You will find out in the practice questions in a moment. First of all, does anyone have any questions? By the way, guys, the first lecture, nucleotide structure, خلصنا. This is all I want you to know about the first lecture. What's the difference between a chromatin and a nucleosome? شوف, a chromatin يعني is basically an umbrella term. One example of chromatin is nucleosome. That's all you need to know, Salah. You know, and the nucleosome will come together or they will form these chromatin loops. Don't go into much detail. This isn't very important, but I mentioned it here. And the nucleosomes are examples of chromatin. That's pretty much it. I'm a bit too lost in the last slide. But one more time, I'll tell you what I want you to know with this. I want you to know in eukaryotes, we have histones. In prokaryotes, we don't have histones. Okay? In eukaryotes, the H1 histone protein is responsible for linking the nucleosomes fibers. In prokaryotes, the DNA is circular. I'm telling you everything I want you to know. Chromatins can be histone and non-histone. Yes, of course, they can be both. But in eukaryotes, what's special is that there is a histone. So H1 just links the histones to DNA? Yes, 100%. The last thing I want you to know is in the nucleosomes, sometime in the question, they won't tell you nucleosome. They will refer to it as a histone octamer. I'm just giving you another name for it. تمام? That's pretty much it. That is pretty much it. How to differentiate viral DNA from other DNA? We will come to it in the DNA replication lecture, inshallah, in a bit. But nothing major that you should know, Salah. طيب, let's move on to this question. Yani, this is literally how they're going to ask you about this. Well, guys, don't attack me for giving you an easy question. This literally came in multiple past midterms. فوالله يمكن you get this question in your midterm. يا رب. من كثر ما إنه it's easy, I genuinely pray for you guys. إنه إن شاء الله they get you this question. And it genuinely came past exams. But it's not like I'm giving you easy questions on purpose. But yeah, everyone's correct. It's the H1 protein. طيب. What about this? What is the name of the structure made of an octameric histone chloride? I just told you guys, and that's basically another name for nucleosomes. Exactly, the answer is C. طيب, we finished the first lecture. I hope it was easy. Everything I said is everything you need to know. Now we're going to move on to the second lecture, DNA replication. It might seem a bit harder, but trust me, guys, wallah, it's much more enjoyable. Much more enjoyable. Nucleosome is the same as H1. لا, look. Nucleosome is made up of histones and DNA. H1 brings the DNA and the histones together, links them together. All of this new structure made up of histones and DNA is called a nucleosome. That's pretty much it. تمام؟ ممتاز. طيب, let's move on to DNA replication. Very fun lecture, guys. Trust me. Inshallah, I'll make it fun for you. First of all, we need to understand that this is a DNA, صح? Two strands. If we want to replicate it, we're going to have to separate the two strands. This is a DNA. We separate it into two parent strands. Why do we call them parent strands? Because these are the strands which will give rise to new daughter strands. Once this DNA separates, every parent strand will copy itself and make a new daughter strand. But in the end result, we will have two new DNAs. Each one of them will compose of one child strand and one parent strand. Is that clear? This is the first baseline concept of DNA replication. We have a DNA double helix, we separate it. Each one of these strands will give a new daughter strand, like here, guys. Let me bring the pointer. This is one double helix DNA. We separated it into two different strands, the ones in red. The ones in red are the parent strands. Each one of these red strands will give a new gray strand, which is called the daughter strand, or the new synthesized strand. But the end result will be two new DNAs, two new double helixes. Each one of those two new double helixes will be composed of one parent strand and one new strand. Is that concept clear, guys? خلاص ممتاز. طيب. The DNA replication, it is the process of duplicating each chromosome before cell division. We're just duplicating the strands. Each new DNA double helix has one original parent strand and one newly made strand, the daughter strand. That's why we call it a semi-conservative replication. Why do we call it semi-conservative? Semi means half, conservative means we're keeping what was in the past. In the past, we had a DNA made up of two strands, but the newly synthesized DNA will have one parent strand and one completely new strand. That's why it's semi-conservative. Half of it is old, or half of it is new. طيب, you guys have a long strand of DNA. But where exactly does the replication begin? Here, or here, or here? The answer to this question is that replication begins at special places. We call them origins of replication. In this origin of replication, the two DNA strands will separate. Let's say the origin of replication is here, then here we won't have any replication. The strand will only separate here, like this. I hope I demonstrated it well. تمام? For these origins of replication are where the replication process happens. Well, I want you to remember, in prokaryotes, best في one origin of replication. And in a DNA strand, we can only have replication in one place, starting in one place. But eukaryotes, they can have 70 million origins of replication. Replication proceeds in both directions. Let's say we have the replication, origin of replication here, then basically the replication will go كذا and كذا, both ways. تمام? If that's clear, we can move on to this slide. Oh, guys, this slide summarizes the whole lecture. I'm going to explain the whole lecture in this slide. وبعدين we're going to talk about everything on its own, which will be very easy. تمام? What is this? It's a DNA strand. Is the origin of replication of the DNA itself? Yes. Somewhere in the DNA, which signals, yalla, time to replicate. You're going to look at it here. This is a DNA molecule. As you guys can see, I want you guys to follow the laser pointer. The laser pointer right now is at the place of the DNA where it's subnormal. We have a double helix, we have two strands, صح? Oh, it's clear in this place, لسه the replication didn't get to it. The replication started from here, from the left side. It's moving like this, cutting through the DNA. But what's clear in that there's something that is cutting the DNA open. As you guys can see, it's like there is a knife, a blade that is cutting the two strands open from left to right. There is an enzyme that does this cutting. We call this enzyme helicase, as you guys can see in the square. Exactly, yes, helicase. The helicase is the enzyme that will open the DNA. The DNA is usually two strands, double helix. We need something to open it first. That will be helicase. طيب, there is another protein That's basically يعني خلينا نقول the assistant of helicase. It helps helicase. It's the vice head of helicase. We call this a single-stranded binding protein. Why do we need it? إنه يا أخي helicase, when it opens the DNA, we need something to stabilize the strands, to keep them in its place, so that the replication process can happen smoothly. We call this a single strand binding proteins. ومن اسمها binding لأنه it binds the strand to keep it in its place. تمام. So we have the helicase opening up the DNA, then the single stranded binding proteins putting the strand in its place. تمام. But there is an issue, guys. The deeper we go into the replication, how can we be sure in those strands that got replicated in zaman won't coil up together will come back into two strands? It's like taking some of your hairs or leaving it again. They will coil up again. We have an enzyme that prevents this from happening, this coiling. And that enzyme is called, yes, Mariam, you're correct, topoisomerase. A topoisomerase will prevent the coiling. فإحنا إلى الآن, what did we do? We got the DNA. We cut it open. We brought the binding protein to hold it in its place. We brought the topoisomerase to prevent it from coiling. خلاص, at this point, nothing on this earth, يعني, استغفر الله, can stop this DNA from staying in its place. But now is the time to actually copy the strands. What is the enzyme that does this copying? DNA polymerase. DNA polymerase is the enzyme that will synthesize the new خلينا نقول DNA. Wait, but how is it moving from left to right? Because there are two strands on the left side. I will explain that in a moment. Just give me a moment of your time. Let's go one by one. Could you repeat topoisomerase? Okay, look. Do you know the string ball that you do this with? But then if you leave it, follows back again. The same thing can happen with the DNA. The more you stretch it, the more you open it, there's a chance in they go back into each other and coil up. Or for example, your hair, if you pull it and then leave it again, it coils back again. We don't want that to happen in the DNA replication process. We want the DNA to stay open like this so that we can replicate it properly. What is going to prevent the coiling from happening? Topoisomerase. Do you guys see it? It's in green. It's making sure that these coils don't happen again. خلاص, is topoisomerase clear? And yes, as we were talking or discussing just now, DNA polymerase, it is the big boss. This is the enzyme that will create the new daughter strand. مو احنا قلنا this is the DNA. It will divide into two. Each strand will give us a new daughter strand. Who is going to be the one to create this new strand? It's the DNA polymerase. The helicase, which opens up, binding proteins, يثبت stabilizes the strand. And topoisomerase, the one that prevents the coiling. These three, what they're doing is they're creating the perfect environment for DNA polymerase to be able to work. Or DNA polymerase will create the new strands. Like now we're going to talk about a very important question and concept actually. I want your full focus with it. Do you guys remember how we talked about in that any strand has a 3' end and a 5' end? يعني هنا مثلا this, the one with, I want you to follow my laser pointer very well. This strand is the parent strand. صح? It's going from 3', as you guys can see, all the way to 5'. صح? طيب. With the one above it. This one, the one here, it's also a parent strand. You guys can see it going all the way here. But this one is going from 5' until 3'. The thing is, DNA polymerase, it can only read the DNA strand from 3 to 5'. يعني DNA polymerase, if you want it to work, take it to the 3' end, which will make its way to the 5' end and read through it. تمام? فإن this strand, this parent strand, which is going from 3' to 5' here, كذا كذا until here, DNA polymerase will start here and it will move until here. from 3 to 5, which is good. It's good. But we have a problem. In this strand, the one above, this is the 3 prime. A DNA polymerase would have to start from right and goes to left. Why is this kind of complicated? لأنه the replication is going this way. Can't you guys see the helicase? The helicase is chopping through the DNA from left to right. But in the strand on the bottom side here, the DNA polymerase, come on, it's moving in the same direction of the helicase as the replication. Everything is going fine. We call this أصلا the leading strand. Then everything is going fine. But in the strand up, the DNA polymerase, it wants to work from the 3 prime to the 5 prime. It's going to start here or goes there. But المشكلة, by doing that, it's going to be going opposite to the helicase enzyme, opposite to the direction of replication. That's why we call it a lagging strand, because it's lagging behind شوية. Is that clear? Is this concept clear, guys? It's a very important question, concept. We will explain it again in future slides, but I want you to understand it now, even if a little bit, لأنه later it will be easy. I will repeat. طيب. شوفوا guys. we have this DNA. We divided it into 2 strands. The helicase is moving like this, صح? Look at it. It's like a knife which is cutting through that side from left to right. تمام? Let's take a look at this parent strand اللي تحت. This one is going from the 3 prime until the 5 prime. صح? While the strand that is on above it, the one on top, it's going from 5 prime until 3 prime. بالعكس. Is the concept until now clear? Is what I just said clear? ممتاز. Yes, Mariam, you're correct. I'll get to that in just a moment. طيب, building up on what I just said, DNA polymerase is a very مرة مزاجي هذا DNA polymerase. DNA polymerase wants to start at the 3 prime of the DNA. Or it wants to go from 3 prime to 5 prime, or else it won't work. It only wants to read the DNA from 3 prime to 5 prime, a DNA polymerase. But in this case, in this parent strand, it's going to come here to the 3 prime, or it's going to go all the way until the 5 prime, and it will create this new strand. Here, in this case, everything is okay. And the DNA polymerase is reading the strand in the same direction as the helicase, as the replication. But in the strand above it, the DNA polymerase هذا, once again, it wants to start from the 3 prime, but it will start from the opposite side, from here. طبعا احنا we're looking at this one, the parent strand, not the daughter, the parent strand. The parent strand, the 3 prime. For the DNA polymerase will start from there all the way until the 5 prime. What's the problem here? In this case, the DNA polymerase is going somewhere, or the helicase with replication is going somewhere else. Do you guys get it now? That's basically the difference in both cases. ولأنه in this strand, they're going in opposite directions, we call it a lagging strand. Logically, the DNA polymerase is going to the opposite of the helicase. A lot of issues will happen. One of these issues is called Okazaki fragments. In the leading strand, everything is good. DNA polymerase and the helicase are going together. They're towards the area of replication. But the strand will be one piece. But the problem is, in the lagging strand, we will have different pieces of new DNA. Fragments basically. We call them Okazaki fragments. تمام? طبعا one more thing about DNA polymerase. DNA polymerase, it likes to start at the 3 prime end, but DNA polymerase copies the DNA template from 3 prime to 5 prime, but synthesizes the new strand from 5 prime to 3 prime. 100%. 100%. We'll get to that in a moment. طيب. Now, this DNA polymerase, يلا, we threw it on the 3 prime end of a DNA. DNA polymerase is a shy person. صح إنه he's very stubborn, or he's demanding to start in the 3 prime end, but he's also a very shy person. He can't start the replicating process unless there is an RNA primer. This primer will stabilize the DNA polymerase in the 3' end, and it will tell it, yalla, this is where you start, no more being shy. But what is going to create this RNA primer? An enzyme called primase. Is this clear? Yani DNA polymerase, it will start in the 3' end, but it needs something to anchor it there. That will be the primer. How can we create a primer? By primase. Last but not least, do you guys remember the Okazaki fragments we talked about? The Okazaki fragments, which are a result of the DNA polymerase going to the opposite of the replication direction. These fragments, يعني في النهاية, they have to join together so they can form one complete strand of DNA. The enzyme that will bring these Okazaki fragments together is called DNA ligase. Ligase because it ligates them together. Tamam? Can you repeat RNA primer and DNA primase? Absolutely. Do you remember DNA polymerase, the enzyme that will synthesize the new strand of DNA? This DNA polymerase will go to the 3' end, but something has to anchor it, stabilize it there. That will be the primer. It's a primer in a sense where it will stabilize it at the 3' end. What is the enzyme that creates or synthesizes this primer? It's going to be the primase. Does it fill the hole? Is that what ligase does? Do you guys see these fragments? This is one Okazaki fragment. This is another Okazaki fragment. This is a third Okazaki fragment. اللي هنا. The DNA ligase will bring them together so they can become one full strand. Tamam, is that clear? Like a magnet, yeah. Tamam, tamam. This is literally the whole lecture. Now we're going to go into a revision phase, خلينا نقول, just to make sure you understand it. For the first step, we said in any DNA replication process, open up the DNA, as you guys can see here. Who opens up the DNA? Helicase. As you guys can see, this green thing is the helicase. It's basically cutting through the DNA, separating the two strands. وبعدين, the single-strand DNA binding proteins, they will come, they will make sure that these two strands are separate or stable, not together again. خلاص? A helicase is a protein that is required to separate and unwind the DNA double helix. With a single-stranded, the DNA binding protein will keep these two strands separate and will stabilize it. Moving on. This is what I meant by coiling, guys. Yeah, basically, when we separate the two strands, keep going, after a period of time, they can coil up again. As I gave you with the example of a curly hair or something like that. But we need topoisomerase because it's going to prevent this twisting and coiling of the DNA. Now, I want you to remember that we have two kinds of topoisomerase. Topoisomerase 1 or topoisomerase 2. Topoisomerase 1, from its name, 1, but it's only going to cleave one strand of the double helix. It's only going to make sure one strand is not coiled. Topoisomerase 2, because it's 2, it's going to cleave the two strands, both of the strands on the double helix. Now, in prokaryotes, topoisomerase 2 has another name. The other name is DNA gyrase. Now, I'm only telling you this because in the exam, maybe they don't tell you topoisomerase 2. Maybe they tell you DNA gyrase. But I just want you to recognize it and, oh, DNA gyrase is basically topoisomerase. Tamam? Last point is that any disruptions in topoisomerase activity, obviously they would lead to DNA damage and cell death. Then if the coiling happens, we can't replicate. We can't have new DNA. What's the significance for it in prokaryotes? Prokaryotes have DNA كمان. يعني prokaryotes, they have DNA. صح إنه they're simpler, they don't have histones, they are circular. But in prokaryotes, DNA is still double-stranded. But they also have replication. Or they serve the same function. تمام? That's pretty much it يعني. Of course, anytime. Right, now let's move on to DNA polymerase. As we said, DNA polymerase, it's responsible for copying the templates or synthesizing the new strands. What did we say? DNA polymerase can only read from the 3 to 5 direction. Yani you take it to the 3 direction, 3', which will move to the 5'. But the new strand it synthesizes will be 5' to 3'. Yani let's go back to this image. This is the DNA polymerase. It's moving from the 3' of the child strand, the parent strand, until the 5 prime. It's reading the parent strand from the 3 prime to the 5 prime. Or the thing is created, the thing that it's creating will be from the 5 prime to the 3 prime. تمام؟ يعني this is the DNA. This is the parent strand of a DNA. Hello? This is the 3 prime, or this is the 5 prime. DNA polymerase will work by going to the 3 prime of the parent or moving until it reaches the 5 prime. What it will create, the result will be, if this is the cap of the highlighter, then the new child strand will look like this. The cap of the highlighter will be on the other side. But the 3 prime of the parent strand will give us the 5 prime of the daughter and new strand, while the 5 prime of the parent strand will give us the 3 prime with this cap of the highlighter of the new strand. Is this clear? So the DNA polymerase will come to the 3 prime, which will move across the parent strand from the 3 prime to the 5 prime. What it's going to create is going to be a new strand that is opposite to the parent strand. For the parent strand 3 to 5, the product will be 3 to 5, 5 to 3. Is that clear? I hope it's clear. طيب ممتاز ممتاز. Okay, let's go back to DNA polymerase. طيب, but this is the function of DNA polymerase. There is just one more function of DNA polymerase. مو احنا قلنا DNA polymerase will create the new strand. يعني it will see the A adenine in the parent, which will give it a T to make the daughter strand. This DNA polymerase, like any human or living thing on the face of Earth, can make mistakes. But DNA polymerase ممكن مرة by accident it puts a C instead of a T. This is why the DNA polymerase has a proofreading mechanism. It's called a 3 to 5 prime exonuclease mechanism. Which makes sense. هو أصلا ال DNA polymerase, it's moving from the 3 prime to the 5 prime. So logically speaking, the proofreading mechanism will also be from a 3 prime to a 5 prime. Exonuclease, it basically will cut off any bases that shouldn't be there, or that are mistakes, basically. Is that clear? This is a function of DNA polymerase I want you to know. But the first function is adding and synthesizing the new strand, with the second function is excise and basically destroy any mismatched nucleotides that were added by accident, basically. طيب, moving on to primase. We said in the primase is the enzyme that's going to give us RNA primer. Why do we need the RNA primer? لأنه the DNA polymerase, it can't start until this primer comes to it, stabilizes it in its place. But the primer comes to the DNA polymerase, puts it in its place, well, that's when the DNA polymerase can start working. Well, this primer is made by something called primase. But this RNA primer is synthesized by the enzyme primase. But the 3 ends of a primer will serve as the starting point for the new DNA strand. As we said, also, the DNA polymerase will only start working from the 3' end anyways. طيب, DNA ligase. Let's go through it fast. We have Okazaki fragments, these fragments that are coming from the lagging strand. طبعا this is a leading strand, it's one piece. This is a lagging strand, it's multiple pieces. We call them Okazaki fragments. But Okazaki fragments are going to come together by the enzyme ligase. But let's read. In one template strand of DNA, the DNA polymerase synthesizing a leading strand that moves towards the replication fork, مع الهليكase, as we said. But the other strand, when we elongate it, as we said, the DNA polymerase will move opposite to the helicase, opposite to the replication area. Well, this will result in Okazaki fragments. Well, these fragments will be joined by DNA ligase to give us one final strand. Is that clear? Okay, we'll go through it once again. I'll go to the same slide that basically explains the whole lecture. We said in Nahina, the parent strand, it's going from 3' until 5'. With the DNA polymerase starts from the 3' end, It's moving to the 5' end. Fortunately, the DNA polymerase is moving in the same direction as the replication, as the helicase. كده, from left to right, from left to right. 3 to 5. The helicase is going from left to right. But in this case, we will have a leading strand. The strand will be normal. But in the strand here, this parent strand, it's going from 5' to 3', which is okay, but we said the DNA polymerase only works from 3 to 5. But the DNA polymerase will come here. It will start here and it will go all the way there. The problem is that when it goes there, it's going in a direction opposite to the helicase. Because of this, we will have fragments forming. We call these fragments Okazaki fragments. These fragments في النهاية, يعني they have to join together to give us one complete single new strand of DNA. What is going to combine these Okazaki fragments? DNA ligase. خلاص, is that clear? Is the concept clear? This is basically what's being said here. And the ligase will get these Okazaki fragments, glue them to each other. ممتاز, ممتاز. طيب, a last very important concept. I think you guys know what telomeres are. Telomere is basically a region at the end of the chromosome. It basically protects it from being degraded. It's like this highlight. Do we ever leave a highlighter like this? لا, لأنه in a matter of time it will dry out or you can't use it anymore or it's dysfunctional. But what if we put this cap? You can leave it for a year, open it again and use it, which will be fine. But this highlighter cap is a telomere. The telomere will go to the end of the DNA, which will cover it to make sure it's protected. That's what a telomere is. تمام? The enzyme telomerase, it protects the telomeres. As simple as that, which prevents them from shortening. So we have a DNA, we have this telomere attached to it to protect it, with the enzyme telomerase will make sure this highlighter cap stays protected. Well, as you guys can see here, without telomeres or telomerases, with each time we are replicating the DNA, the new strand will get shorter, وبعدين shorter. But that's why we need telomerases, to make sure we have the perfect size of a strand. طبعا, something I want you to know. You guys know what stem cells are. They divide, they give rise to our organs. Our reproductive cells, sperm cells, egg cells, every month, every day, they're proliferating. In this case, that enzyme telomerase is very active because we want a lot of divisions. If we didn't want a lot of divisions, خلاص, remove the telomerase so that the cell can slowly get smaller and smaller and dies. But in this case, we need the telomerase. طيب, most body cells, we have low telomerases. So the telomeres shorten with age, or the cells eventually stop dividing or even die. Well, this is why we age أصلا. يعني, this is why every human dies in the end. لأنه as days and months and years pass by, والله يحمينا ويطول لنا أعمارنا جميعا بالخير. Our DNA is replicating more and more and more, and each time it's getting shorter and shorter and shorter until خلاص. Lastly, we have cancer cells. We know in the cancer cells, they grow at a very incredible and scary rate. They grow and grow and grow and they don't die. In this case, the telomerase is reactive. The telomerase, it's basically providing the cancer cells with immortality, preventing them from ever dying. Last but not least, I want you to know, and obviously, logically, longer telomeres would mean more survival, longer survival. خلاص, is this concept clear? So we're almost done with the lecture. 80% done. So just tell me if this concept is clear or we can move on. Why can't we make telomeres? Yani I think we would be immortal in this case. And yes, I'm pretty sure the scientists have experimented on this. I heard about this before, and that they tried to maintain the telomeres or create new ones, but I don't think anything can beat the undeniable fate of humans dying. Sorry, but I can't get primase and primer. Okay, no worries. We'll go through each one more time. Look, we said in a DNA polymerase, it will go to the 3' end. This is the DNA polymerase. Goes to the 3' end, which will synthesize a new strand. DNA polymerase, the lure. Okay? He's sensitive, and someone has to really put him in his place for him to do his job. That's why we need the primer. The primer will stabilize the DNA polymerase, and it will be like, do your job. This is not the time to mess around. Bas. طيب, with this RNA primer, أكيد it's not going to spawn out of nowhere. Something has to create this primer. What creates the primer is the primase. As simple as that. Is that clear? Of course. طيب, let's go to one last concept. You guys asked me about viruses, صح? Viruses ما عندهم DNA polymerase. They have RNA polymerase. طيب, there is one main difference between DNA polymerase and RNA polymerase. I want someone in the chat to tell me what were the two functions, not one, two functions of DNA polymerase. I want the functions of DNA polymerase. What does DNA polymerase do? That's the question. Exactly, DNA replication and proofreading. ممتاز. طيب, the thing is, in RNA polymerase, we don't have proofreading. يعني A can randomly have a G, C can randomly have a U, لخبطة. But why does this concern us? لأنه each time the virus replicates, it's like a new strand is being created for more mutations. When we have more mutations, this is why it's hard to make a vaccine. لأنه every time we make a vaccine, we realize that the virus changed. It has a new strand. We have to make another vaccine for it. Is the concept clear? If it's clear, then we have made it to this checkpoint. Well, I would like to take any questions, yeah, if you guys have any. طيب, I'm assuming إنه إن شاء الله everything is clear. Now we can move on to the practice questions. I'd love to receive your answers. Is there one more lecture? لا 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 خلاص, we're almost done. Wallahi, we're almost done. We are 85% done. 87% had to be specific. So in viruses, they have polymerase, but without proofreading. Yes, and because they don't have proofreading, mutations will always happen. Because mutations happen, it's hard to find a vaccine. We'll create one, yeah. طيب guys, يلا, more people answered. We only have 3 answers. This should be an easy question. Exactly, the answer is B. Very simple question. Which enzyme is responsible for inhibiting supercoiling? The twisting, قلنا, the supercoiling. It's topoisomerase. As simple as that. تمام? يلا, next question. Give me your answer. Exactly, 100%. The answer of A. تمام? The telomeres are getting shorter. What's happening? What's the abnormality? And we can't have extensions of the telomeres. The strands are getting shorter. طيب, this question. Are these past questions? Yeah, most of these questions are past questions, even if not the exact same in terms of wording, it's the exact same concept. فوالله العظيم, 50% of, if not more, if not all of the questions I brought today, they will be the same ones you see in your exam. So telomeres can only be found on the 3' end. Not necessarily. لا, بس هنا يعني in the answer it just happened to be 3' end. بس it could be anywhere. طيب يلا, this question guys, it's quite tricky. It's telling you this is the parent strand. What will be the product? To answer this question, I want you to imagine yourself, you, my dear first year, as the DNA polymerase. Yalla, now you guys are the DNA polymerase. Where will you start? هنا ولا هنا? Tell me. If you were the DNA polymerase. From 3, exactly. طيب, خلاص, now it's simple. T, A, G, C, C, G, A, T. Well, as you guys all answered correctly, the answer will be D. Is it clear? Are we on the same page? ممتاز. Okay. يلا. It's basically asking you why is it difficult to create a viral vaccine? Okay, exactly. The answer to this question will be A. Okay, يلا خلاص. The last 3 slides of the lecture وبعدين we'll be done inshallah. We're gonna talk about the pharmacology aspect. A lot of weird drugs, weird names. But wallahi guys, it's important. They get you memorization questions from this. But just focus with me on this stage. There are topoisomerase inhibitors. But some of you might ask me, why do we want to ever inhibit topoisomerase? We want DNA replication. My answer is 2 cases. If you have bacterial cells in your body that are causing a disease, you want to kill them. But that's one way, or one, let's say, use. The second use, as you guys just said, Abdullah Muhammad, it's for cancer cells, to kill the cancer cells. That's when we want to use topoisomerase inhibitors. When we inhibit topoisomerase, خلاص, the DNA will coil together, no more new DNA, cells die. Now I want you to memorize the drugs that inhibit topoisomerase 2. The prokaryotic ones, which are basically antibiotics, we use them for bacteria, we call them fluoroquinolones. You will take this in CVP next year, you'll remember it, but I just want you to remember it now. It's very important. Well, I want you to remember the 2 examples, ciprofloxacin and levofloxacin. Anything that ends with floxacin, basically. Anything that ends with floxacin is a topoisomerase 2 inhibitor. خلاص? Well, these are the prokaryotic ones. But basically, they target bacteria, these stuff, antibiotics. The eukaryotic topoisomerase 2 inhibitors, the important ones are daunorubicin and doxorubicin. Anything that ends with rubicin. خلاص? If you see one of those in your exam, خلاص, link it to topoisomerase 2 inhibitors. It's used to promote the coiling to kill either cancer cells or bacterial cells. طيب. Another thing, nucleoside analogs. Do you guys know how إنه مثلا if we have an A in a parent strand? I'll ask you this question. What will be the complementary base in the daughter strand? If we have an A in the parent, what will be in the complementary strand? Exactly, T. طيب, nucleoside analogs is like a special form of T. We inject it into something to stop the DNA replication. حلو؟ يعني مثلا احنا we have this is A. This is adenine. Adenine obviously will bind to mean, as you guys said, thymine. A thymine analog is something that looks exactly like thymine. They're both highlighters. But the point is, we want the fake thymine to go with the adenine. Once the fake one goes to the adenine, it will ruin everything. خلاص, is the concept of analogs clear? Analog means similar. Nucleoside analogs means things that are similar to the nucleoside. This is, for example, cytosine. See, we know when the cytosine binds with ash, guanine, which is this. A guanine analog is a structure that looks exactly like guanine, but it will trick the original strand into thinking it's the actual guanine, which will bind with it. في النهاية, this is the analog, it's not the real guanine, but it will block the DNA elongation, which will prevent any further replication. Is it clear now, guys? Well, I want you to remember 2 of them, zidovudine and didanosine. Just remember, those 2 are nucleoside analogs. Will they ask about drugs not in red? I've never seen a drug not in red in the exam or in any past papers. I scanned through them. Just remember them. I put them in the slides لأنه أكيد they're important, The V, if you had to remember some of them, it's the ones in red. Ideally, I want you to remember all of them, okay? I'll repeat the concept of nucleoside analogs one last time. We have thymine. This is a thymine molecule, تمام? We know when the thymine binds with adenine, T and A. But this is adenine. Usually thymine will bind to the adenine. An adenine analog is a structure that looks exactly like adenine. Look, they're both highlighters. They look like each other. The point of using an analog is that we want to trick the original strand into thinking it's the actual خلينا نقول base and bind with it. This will result in the termination of the DNA replication process. ليش؟ لأنه this is not the actual adenine. It's an analog. It just looks like the actual one. So basically, nucleoside analogs, what they do is they catfish the parent strand. It's a catfishing process, basically. It's a bait, or more often than not, the parent strand takes the bait. The catfishing works. خلاص, is the concept clear, guys? ممتاز ممتاز. طيب, now we're going to talk about DNA repair. We said إنه the DNA polymerase always proofreads while it's synthesizing the new pair. Sometimes, even with the proofreading, غلطة تصير. We have a mismatch. The DNA polymerase finishes its job, ولسه there is a mistake. This is why we need DNA repair. How does this happen? Basically, a mismatch repair system. This mismatch repair system will fix any stuff that are wrong after the replication process finished. يعني احنا أول شيء we have DNA polymerase proofreading, after it, if there are mistakes, the mismatch repair system will take over. طيب, what if someone has a mutation in the mismatch repair system? They will have Lynch syndrome, which is a form of colorectal cancer. This is very important for you to know. I want you to memorize this. Lynch syndrome equals problems in the mismatch repair system. Yes, proofreading occurs. Okay, guys, you're writing an essay for your English elective. When you proofread your own essay, it's before you submit it to the teacher or to the doctor. صح? خلاص, you just wrote the essay, you're proofreading it yourself, then you submit it. Mismatch repair is when the doctor reads your essay. It's after you tried proofreading it. Or sometimes, even when we proofread, we still make mistakes. صح? That's when the doctor, the mismatch repair system, comes in, gets involved, and fixes it for you. Is the concept clear now? Or if we have a problem in this mismatch repair system, it will be associated with Lynch syndrome, which is a form of colorectal cancer. خلاص, is this slide clear? Exactly, Mohamad. If the doctor doesn't get the issue in your paper that you proofread, then this is Lynch syndrome. Yeah, exactly. طيب. A very specific issue that happens in DNA is thymine, with thymine. مشكلة thymine, الله يهديه, thymine is very freaky. Whenever two thymines are next to each other, they immediately form dimers. They connect to each other. This is very bad, and that disrupts the whole DNA. Hello? UV lights, ultraviolet lights, when someone is exposed to them, they can cause these thymines to get freaky or connect with each other. Well, this is a problem, and it blocks replication, it blocks gene expression. We have something called a nucleotide excision repair system. This nucleotide excision repair system is going to remove the thymine dimers. We don't want the dimers from each other. So if we have a problem in this repair pathway, the dimers will stay together. We will have a disease called xeroderma pigmentosum. It's caused by mutations in the nucleotide excision repair system. Happens only with thymine? لا. It happens with pyrimidines in general. Pyrimidines. اللي هم one ring. That's most likely 80% of the time, it's with thymine. تمام? In patients with xeroderma pigmentosum, they basically don't have the nucleotide excision repair system. Basically, when the UV light strikes this person, the thymines will come together, or nothing will separate them. Well, this is a huge issue. With xeroderma pigmentosum leads to UV sensitivity and high risk of skin cancer, as you guys can see in this poor child. خلاص? With that, نكون خلصنا lecture. This is the final checkpoint. Let me know if you have any questions so we can move on to the practice questions. Sorry for the dumb question. No question is dumb. Wallahi, we're all here to learn. There is no shame in any question. If telomerase helps for long cell lifespan, why we could not estimate the death of the patient by the length of the telomerase? Okay, look, I'll tell you something. Telomerase, يعني rarely ever does a person die because the telomerase خلاص got too short. Most people we know died of accidents, cancers, diseases. صح? All of these are not related to the telomerase. Telomerase is basically in an ideal, perfect world where there are no diseases, where the only thing that will cause you to die is this telomeres. That's when we can do this, you know? But I'm pretty sure in an actual clinical practice, they try to estimate يعني, the lifespan. It's a normal death, yeah, pretty much. Can you explain the thymine dimers again, please? Of course. Okay. Pyrimidions in specific zero pigmentosum is caused by T connecting to each other, forming a diamond because of the UV rays. So is there something else I need to know? No, that's pretty much it. You just need to know that thymine dimers, usually in a normal person, they're disconnected by the nucleotide excision repair. In patients of xeroderma pigmentosum ما عندهم this excision repair. تمام؟ طيب, for the person that asked, look, pyrimidines, most specifically thymine, um, they always connect to each other whenever they have the chance. This is bad. Thymine المفروض يقعد مع الأدينين والثymine المفروض يقعد مع الأدينين. But sometimes these thymines come together, like here in this picture. فيصير الأدينينز قاعدين لحالهم. They're not binding to each other. والthymines, for some reason, they're together. You know? Um, so basically, this is an issue, then it will basically cause a blockage in the DNA replication and gene expression. تمام? But usually, whenever this thing happens, when there is a connection between the two thymine dimers, our body naturally stops it by a system called a nucleotide excision repair system. It's found in all of us, alhamdulillah. During severe UV exposure, لا. Maybe, probably this happens in our bodies hundreds of times a day when we walk under the sun. But لأنه we have a normal excision repair system, alhamdulillah, nothing happens to us. But in patients of xeroderma pigmentosum, they don't have this excision repair system. They don't have it. So any exposure to the sun with the UV rays will make the thymines come together. وبعدين there is no excision repair system to remove them. Everything becomes bad. These patients always have high UV sensitivity and high risk of skin cancer. House, is everything clear? Do we have any other questions about the lecture? ممتاز, alhamdulillah. طيب, let's move on to the final few practice questions وبعدين we're officially done. Yalla, let me know what you guys think the answer is. Of course, wallahi, anytime. I try my best to simplify everything. Inshallah, this will be more than enough for your midterm and final. Absolutely, you guys are correct. Mismatch repair. طيب, if the answer were to be A, what would the question tell you? Xeroderma pigmentosum. Ya salam alaykum. طب, let's say the question didn't tell you it's xeroderma pigmentosum. They gave you the clinical signs, how the patient would present, how they would look like. How would you describe someone with xeroderma pigmentosum? Exactly, hypersensitivity to UV, can't be exposed to the sun, high risk of skin cancer, mashallah alayk. طيب, yalla, this question, the memorization question. It's pure memorization. Rubicin, we talked about this in detail. Exactly, C, topoisomerase 2. لا, not D, not topoisomerase 1, topoisomerase 2. If we go back to the table, it's topoisomerase 2 inhibitors. هذولي doxorubicin, daunorubicin, ciprofloxacin, levofloxacin. تمام؟ ممتاز. طيب, what about this question? Yeah, you have to memorize them, wallah. The analogs, the nucleoside analogs, and the topoisomerase 2 inhibitors. Yalla, guys, give me an answer to this question. It's the last question, by the way, of the lecture. Khalas. Yalla, everyone answer it. I want everyone to answer. No, we're literally done. Okay, I'm seeing a lot of C's. MashaAllah alaykum. The answer is, in fact, C, removal of pyrimidine dimers. Khalas, I hope the lecture is clear. Thank you guys for joining. Wallah, this is more than enough for your midterm. I also have another few PAL sessions for MOL 1 from last year. I recommend it to you guys, not just because I gave the sessions, but wallah, they always get questions from the same concepts I'm talking about, so make sure to visit those PAL sessions. Once again, Allah yafikkum. Inshallah, the MOL midterm will be very easy for you guys. Obviously, if you have any questions, never hesitate to contact me ever. I'm always here for you guys, wallah. What matters most is you succeeding or you doing well and succeeding on your exams will make my day, wallah. If not my year. Salaha. Can we study only from your slides for the two lectures? Yes. Look, any PAL session I do just depends on the PAL session or you'll do well. Khalas? Does anyone have any questions? Abdullah, we can stop the recording if you want. I'll just stay for a few more minutes to take questions if anyone has any questions.